Okay, so during lactotin, first the nuclear membrane becomes thick, prominent, grows. Second, the chromatin material, which is a thread-like structure, it will condense, shorten, and will be changed into chromosomes. So what happens in lactotin? We discussed three changes. Number one, this nuclear membrane appears, it becomes thick and prominent, grows. Second, we discuss the appearance of chromatin material, it becomes prominent. Thirdly, it shortens, it will be shrinked and change into chromosomes. When like it will be now in this form, it will be changed into chromosomes. Basically over here it will be a single structure chromosome. Next, let's discuss what will happen in zygote. In zygote, now the process will start of finding pairs. The chromosome number 1 from maternal side will find its pair, that is from maternal side. So paternal and maternal pair 1 will get close together. So first pair will be formed. So similarly second, third, fourth, fifth, all 23 pairs will be formed. So these chromosomes are actually same in their structure, shape, size, amount of genetic material. So that's why they are called homologous chromosomes. So when we talk about the process of pairing of homologous chromosomes, this process is called synapsis. Synapsis is actually the pairing of chromosomes, mean one from maternal and one from paternal side. So in this way all the 23 sets will be arranged. This arrangement of chromosomes or lining up of chromosomes is called synapsis. But one difference that we will discuss, when the chromosomes will shift in their shapes from lactotin to zygote, over here they are single structured chromosomes, but when they reach here, now they are double structured chromosomes. Why they are double structured chromosomes? Actually, the amount of genetic material is going to be replicated. DNA is going to be replicated. When DNA is replicated, over here it was single structured chromosome, now it becomes double structured chromosome, double the amount of genetic material. So in zygote, again recap the changes. Number one, the homologous chromosomes pairs up. This pairing is called synapsis. First they were single structured chromosomes, but now they have been changed into double structured chromosomes. Mean each chromosome first was with one chromatid, right now it is provided with two chromatids, and here it will be called a tetrad or bivalent structure. Tetrad or bivalent. And we discussed two chromatids in same chromosome are sister chromatids, but with the position of other chromosome they will be non-sister chromatids. After this change we move to next, the pachytene. When we talk about the next subphase, pachytene, during this pachytene these chromosomes will become more condensed and a unique process will take place over here. In this phase the non-sister homology, the chromatids, they starts overlapping. How they will overlap? Like this is a chromosome, from maternal side, and this one is a chromosome from paternal side. So maternal and paternal side chromosomes, they starts overlapping, their chromatids starts overlapping. During this overlap they make chiasmata connections. The overlapping sides, the positions where they overlap, they make chiasmata. So these are the junctions, these are the links where these chromatids or the chromosomes will reshuffle their genetic material, they will interchange their genetic material. This is the reason that will bring some variations. This is the reason the siblings in a family, they have variations in their height, in their intelligence, in their features. So we are not same, even we are siblings, belongs to same father and mother, we have different variations. Even the Siamese twins, the identical twins are not 100% same. What is the reason? This is the pachytene stage, because during this stage two different types of chromosomes, one from the male side, the paternal side, and second from the maternal or female side. When these two type of chromatids or chromosomes, when they overlap, they exchange their genetic material. When they exchange their genetic material, though keep in your mind, they are homologous, it means they will be having same positions of gene locus. If we are having genes for eye color over here at paternal side, the same genes will be present at same position on maternal chromosome. If we are having a gene for the shape of the nose, same gene at same position will be present in the paternal side or paternal chromosome. So this is the reason these genes, these genetic materials are going to be reshuffled, they are going to be changed, and these changes bring a lot of differentiations in the next generation. So during this process, when genes are exchanged, this process is called crossing over. So this process will take place during pachytene. Then we enter into next phase, that is called diplotin. During this diplotin, these chromatids which are overlapped at chiasmata positions, what will happen? These positions repel now these chromosomes to opposite sides. When they are pulling up, or their chromatids are pulling away from each other. So in this way, when we will enter into diakinesis. Diakinesis, when the chromosomes will be separated, they will be disjuncted. When disjunction will take place, they will be separated from each other. So over there we will observe one change. What is that? A part of this chromosome now is there with changed genetic material, and same is the case with this chromosome. So this is a result of crossing over. So this was all about the process of prophase 1. Students, in our next lesson I will recap all these changes, and in detail I will tell you about the prophase 1, that what changes actually take place during the process of prophase 1. Till then, stay happy and blessed.